Hi, I'm Jake Glanville, Chief Science Officer of Distributed Bio. We're the creators of Abgenesis, an advanced antibody analysis platform. What I'm going to do today is show you a quick example of how our Abgenesis can accelerate antibody repertoire analysis. I'm going to show you a research study that I conducted in 2010 and 2011, where we looked at identical twins and the genetic heritability of their immune systems. That study took me over a year to complete, and it was ultimately published in PNAS. I'm going to show you the results of that study, and then we're going to see how I can upload that same data set and within a matter of hours come to the same conclusions using the Abgenesis platform. The study had a pretty simple design and a nice set of conclusions. What we did is we took 4 individuals. Each of the pairs were identical twins, and from each one of the individuals we got 2 parallel blood draws. In addition, we knew that one of the twin pairs had a patient, twin B1, who had been afflicted by multiple sclerosis for over a 20-year disease course. We took those samples and we provided them in a blinded manner. So I had the blood, I knew they were replicates, but I didn't know which twin, which individual, or which replicate that particular sample was. And we did antibody sequencing blind. And the reason we did this is we wanted to ask 3 important questions. First, could we tell the twins apart? That is, could we distinguish antibody repertoires based on the underlying genotype? Second, would it be possible just by looking at the antibodies to distinguish identical twins? And finally, do you think it would be possible looking just at the antibodies to identify the sick twin of all the samples that we analyzed? So what did we find? Our first observation was that we could definitely tell twins apart just based on their naive V gene usage profiles. We find that the twin pairs use the V segments in a rate which is dictated by the genotype and is strong enough in its differences to be able to reliably distinguish identical twins just by looking at their antibody repertoires. Surprisingly, even though twins are using their V genes at the same rates, the antibodies that they produce are almost entirely personal. So if I had an evil twin, Bizarro Jake, he and I would use our V genes at the same rates on our cells, but we'd produce entirely unique populations of antibodies in response to our environment. Finally, we noticed that one set of samples looked very different than the rest. And the difference was that it had entirely lost its memory compartment. All of the samples looked like they were naive cells, and that was a result recapitulated in CD27 positive flow cytometry sorting. That turned out to be the affected twin, and it looks like what we were seeing was not so much the effect of the disease, but rather the effect of the treatment, 20 years of lymphocyte ablation and immunosuppressive therapy. That study took over a year to produce. Let's go ahead now and see how quickly we're able to reach those same conclusions using that same data set with Abgenesis. We'll begin by logging in. Abgenesis is a web-based framework that allows you to analyze Sanger sequencing as well as high-throughput sequencing data. It has a projects environment, a sets of experiments, and a collection of analysis apps that can be used to perform additional repertoire characterization. Let's go ahead and add an experiment. The Add an Experiment page asks us a couple questions. I'm going to go ahead and say I want the project to be in my test project. I'm going to name the experiment Twin Study 2011. The description is rerun the twins. It's going to ask me if I want to run antibody lineages. I'm going to say yes. That was an important feature of being able to distinguish the twins by normalizing out recent immune events. Asked, do I want to run developability? This is asking would I want to turn any of those antibodies into a drug. I don't, but for the purpose of the demonstration, let's just include it so we can perform additional analysis of features that might be relevant for drug development. It'll tell us some interesting things about in-link glycosylation, acid hydrolysis, deamination sites, and germlining of framework residues. I'd like to see 100 top lineages to display and 100 top clones. Keep in mind we're uploading a few million sequences, so I don't really want to have them all in the report, but they'll be available to me later for download and manipulation of the total data set. And that's it. Now I can go ahead and upload those sequences. And here they are. We'll just drag to select. Go ahead and click Open. And those samples begin to upload into the environment. Abgenesis can either be installed in your local network, or we can host it for you on the Amazon cloud. As each sample gets uploaded, we see a single tab appears, allowing us to be able to assign a series of annotations for that sample. These are just for your convenience. You're storing the species, human, the isotype, it's a mixture, so we'll say other. I can change the sample name. I'm just going to call this mid1. Maybe I'll assign it here as well. The phenotype is healthy, presumably, and I can assign a time point. It'll take Abgenesis an hour or two to complete the analysis. Instead of spending a year on research, I'll go grab a coffee. Alright, when it completes, you see it show up here in the Experiments tab. Twins, the QC status is success. Let's go ahead and give that project a click, and that's going to take you to the experiment page. The experiment page organizes my results in a series of tabs so that I'm not overwhelmed by all the types of analysis that the Abgenesis platform produces. The first tab, Experiment, first tells me the parameters that I set when I ran this experiment and the collection of samples that were uploaded. Let's go straight to the Analysis tab. The goal of the Analysis tab is to perform a series of sample-wide QC across the sequences that were uploaded. Right here in read length distribution, you can immediately see that there's two different read lengths, and this is a function of the efficiency of some of the PCRs. In the read depth distribution, it tells me that I achieved at least 50,000 reads, if not more so, across every single sample, that I was able to cover heavy chains, kappa chains, lambda chains, and that there weren't too many off-target sequences in each sample. Read orientation assures me that I received both forward and reverse reads in roughly the same proportions. An analysis of cross-sample clones asks for every file that I uploaded, which other files share common antibodies. And you can see that there are basically 4 bricks that are created here. What's happening is that this is gathering a set of mid-7, mid-4, mid-1, and mid-10. And those are the barcodes for the 4 different twins. The double groupings you see here reflect groupings of technical replicates and groupings of biological replicates within individuals. This is basically recapitulating the research that was performed in the study, showing that people produce mostly individual antibodies with very little overlap. Next, when we look at the somatic hypermutation profiles for every single sample, we find that there's a subset of samples that have a very low amount of somatic hypermutation across all of those samples. This is the afflicted twin. That's the second piece of research that we recovered from the study, and now it's being provided as a default output, showing that those samples are very unlike all of the rest. Okay, so we're finished looking at QC. Let's look at the Antibodies tab. In the Antibodies tab, now for each sample that I uploaded, I get a rich distribution of V segment usage, J segment usage, CDR3 distribution, and then clonal expansion, number of reads per clone, shown across the families observed. I'm going to scroll down and take a look at our afflicted twin compared to some of the healthy individuals. In a healthy twin, I can see a broad variety of clonal expansions happening in their immune compartment, a wide distribution of V segments that I can export and then compare across samples, and a nice distribution of CDRH3s and J segments. When I look at their individual antibodies, I can find a tabular layout of the most enriched antibodies, so these are the things that had expanded most in their immune system most recently, the percent germline identity of all of those clones, and the translated CDRs. By clicking around here, I can look at the most conserved to the most mutated molecules. When I compare this healthy twin to one of the afflicted twin members, I find a notable suppression of clonal expansion. It looks like there's a couple of members that have clonally expanded, and this axis is notably altered in the number of sequences I've seen, with the vast majority of reads showing up only once. And when I look at this person's individual antibodies, sorting by germline, I find that the most dominant clones in this person that showed up many times look like they have some affinity maturation, but essentially everything else is almost entirely germline, down to probably the read error rate of the high throughput sequencing. This both doubly confirms what we saw before about the somatic hypermutation, and it also gives us the ability to go in and interrogate these specific molecules more later, in case we're interested to see what it is that makes these particular recent expansions special. Maybe if it's disease relevant or relevant for a recent immune event. Note that I can copy any one of these CDR3s, and Abgenesis provides me a mechanism to search the entire system for any examples where that molecule has been found previously. So if I search with the CDRH3, it takes me back to this particular experiment and I can go explore. And that's by identical match. But you can imagine you may want to do something more sophisticated. Knowing that not every question you might want to ask can be preloaded into a report, we also created an Analysis Apps tab. This Analysis Apps tab makes it extremely easy to add new types of analysis into the environment to enrich your ability to ask questions within experiments, and across all the projects. We perform BLAST, profiling, pairwise comparisons, and really extend any new type of bioinformatics that we will do and spread out to our users, and you can do internally and share your bioinformatics with others, creating an ecology of different computational needs to be able to address the ever-changing requirements of antibody repertoire analysis. Thank you for your time in looking at how Abgenesis can analyze something that took me a year now in under an hour. The question becomes, what do you want to analyze next?